Hello, I'm Hannah Donnert with the Collaborative on Health and the Environment. Che enjoys bringing you the latest environmental health science through our partnership calls and webinars, science serves, publications, and social media. I would like to welcome everyone to today's Che EDC Strategies Partnership webinar, which is titled Consensus on the Key Characteristics of Endocrine-disrupting Chemicals as a Basis for Hazard Identification. Our moderator today is Jerry Heindel, founder and director of Commonwealth's Healthy Environment and Endocrine-disrupting Strategies. We will leave time following the presentation for a brief Q&A session. You may type in questions through the Q&A feature available on the menu bar at the top of your window at any point during the presentation. After the presentation, our moderator will read out questions for our speaker to respond to. We will get to as many comments and questions as we can during the Q&A period. For those of you who called in on the phone, we have posted slides to accompany today's webinar on our website. You can download these by going to healthandenvironment.org. Please scroll to the bottom of the page and select today's webinar. On the webinar page is a link to the slides. Everyone on the webinar right now is muted with the exception of our moderator and our speaker. This webinar is scheduled to last for 30 minutes and is being recorded for our call and webinar archive. With that, I'll turn things over to you, Jerry.
Okay, thank you very much and welcome everybody to today's webinar. As you heard, our speaker today is Dr. Michelle LaMerrill. Dr. LaMerrill earned her PhD in toxicology just down the street here at the University of North Carolina, Chapel Hill, and her MPH in epidemiology at Mount Sinai School of Medicine. She's currently an associate professor of environmental toxicology at the University of California, Davis. She received the WUNS Award from NIEHS in 2016. And as you all know, the WUNS Award is a very prestigious grant for young investigators. Her research program is quite unique. She uses cellular, whole animal, and human epidemiological studies to link toxicological evidence of mechanisms of adverse health effects to human environmental health research. She's particularly focused on the associations between exposure to environmental pollutants and the risk of endocrine diseases, obesity, diabetes, hypertension, and breast cancer. Today, she's going to discuss her recent paper on the consensus of key characteristics of endocrine-disrupting chemicals as a basis for hazard identification. As you will see, this paper is a very important addition to the endocrine disruptor field. So, I turn the webinar now over to you, Michelle.
Can you hear me?
Yes.
Great. All right, let's get started. First, I would like to thank you, Jerry and Hannah, as part of the Che group to invite me for this webinar. So, for those of you that are not familiar with endocrine disrupting chemicals, I'd like to start with a brief definition, and this is a definition provided by the Endocrine Society. There is no international consensus on what an EDC is. However, the Endocrine Society, being a professional organization of medical doctors and academic researchers, I think is the authority on it from a real scientific perspective. And they say that an endocrine disrupting chemical is an exogenous, or what we call non-natural chemical, or a mixture of chemicals that interferes with any aspect of hormone action. And this was defined in the position statement from the Endocrine Society that is in the bottom part of the slide. So now that we know what EDCs are, how can we identify them? So one of the things that we need to think about in terms of identification is kind of a jargony word, which is hazard. And when we're working as societies to identify EDCs, we think about them in terms of hazards, which is really this intrinsic property or characteristic that is embodied by a chemical and can make that substance dangerous. And so with EDCs, we currently have a very complex, rich literature of mechanistic experimental studies. And they clearly demonstrate these endocrine disrupting hazards, so these properties of the chemicals. But there's not a widely accepted systematic way to integrate these data to help different organizations around the world identify the EDCs. There are other hazardous substances that have worked to do identification, and that is with relationship to carcinogens and the International Agency for Research on Cancer, also known as IARC. So IARC is part of the World Health Organization, and they're kind of the world expert to define a chemical as a carcinogen. And so they've developed a method that's clear and reproducible that they use regularly to identify carcinogens. Previously, they relied more heavily on epidemiology and rodent studies, like the official rodent studies that the National Toxicology Program of the United States conducts. But over time, the number of these kinds of studies are being reduced. People don't want to have animal testing, and the epidemiology studies of cancer are quite expensive to conduct and are just not being funded in the same ways they used to be. At the same time that those are decreasing, we're seeing an increase in more mechanistic experimental studies and also in vitro studies. And so there's this trend of a shift where if you look at the diagram on the top, the IARC framework is to integrate the human studies or the epidemiology studies, those rodent or animal phenotypes, the animals getting cancer, and then all of the mechanistic data. But as we see mechanism playing a larger part, we need to think about how to organize and evaluate that mechanism better. And so out of that effort, IARC came up with the key characteristics of carcinogens. You can see a snapshot of the original article about that in the bottom corner. And this characteristics framework is for organizing the data related to those intrinsic properties that are revealed by experimental studies. And what's great about them is because we're looking at them in terms of properties, if we have an incomplete mechanistic pathway, that doesn't prevent us from decision making. And one of the things that makes that so important is we seldom know the whole pathway of how something causes a disease such as cancer. So even cigarette smoking has been studied very extensively, but scientists who are rigorous would have a hard time drawing definitive arrows from one step to the next. And because of that, even with a well-known substance interfering with our ability to make a complete pathway with arrows, what that does is it interferes with decision making. And so this characteristics framework instead allows us to not be kind of paralyzed by these arrow systems. This is also great because you're populating your exploration with these characteristics, and that helps identify data gaps. So looking at that framework that the IARC developed, the CalEPA wondered if we could develop key characteristics approaches to other types of hazards besides carcinogens. And so in 2018, a meeting was convened in Berkeley where Tom Zoeller and myself were invited to lead the evaluation of whether or not it was feasible to develop some key characteristics framework for the EDCs. Now that has just been published in the last few weeks, and you can see a snapshot of that. It's a free article available for download. So what are these properties of the endocrine disrupting chemicals? So if you look in the top left, you can see the first one. The numbering is a bit arbitrary, although loosely the first half of them relate to the receptors that hormones bind, and the second half more or less relates to the hormones themselves. So properties of endocrine disrupting chemicals is that they're able to bind receptors and activate them or be agonists. Another property is that they can actually antagonize the receptors or inhibit the receptor's downstream function. The third characteristic is modifying the hormone receptor expression. Because the hormone's action is dictated by that binding to the receptor, if there's not as many receptors present, or too many receptors present, that can modify the hormone action and disrupt the endocrine system. Another aspect of a characteristic of EDCs is called signal transduction, and this is really the actions that take place downstream of that receptor activation. And so depending on whether a receptor is activated or inhibited, the signals that it sends can be amplified or muted, and of course that would disrupt endocrine function. Another aspect of a key characteristic of an EDC is altering the epigenetic system. And so epigenetic system includes methylation of DNA, for example, modifications of histones, the proteins that help organize the packaging and access to DNA, and also these RNA that are involved in regulations. And so epigenetic alterations are some of the ways that our bodies create regulation of genes that help with signal transduction, more or less. So those are a lot of the properties that relate to some receptor functions. Some of them can also relate directly to the hormones, but specifically now we'll look through the next couple of KCs, which really relate to kind of the hormone life cycle, if you will. So the sixth key characteristic of an EDC is something that which can modify the synthesis of a hormone. So you can imagine having too much or too little, or some hormone produced at the wrong time, really disrupting the hormone system. Similarly, on KC7, you can imagine hormone transport, so the movement of a hormone from where it's produced into the bloodstream or perhaps into the target tissue also have very important implications for the function of the hormone system. In 8, you're looking at the key characteristic of influencing where the hormone ends up. How much of that hormone is in your blood? How much of it is in your target tissue? Another characteristic of EDCs is influencing how that hormone is metabolized or broken down or cleared from our blood. So hormones have very specific actions that are supposed to happen at very specific times, and it's just as important to get rid of them when we want to get rid of them as it is to make them and move them around. So this is also a key characteristic of EDCs when that's modified. The last key characteristic is 10 for fate, and this is really an important one for the developmental susceptibility to endocrine disruptors, where we're looking at the fate of cells, so how much they're proliferating or perhaps dying or turning into one cell type instead of another. So what can we do with this framework of KCs of EDCs? We have some recommended uses, and people aren't limited to these uses, but to start the conversation, we think it's helpful to go over them. You can use the KCs of EDCs to search and organize the relevant literature. And this is one way in which the KCs of carcinogens have been a huge help to IARC. Searching and organizing the literature in a systematic way using specific key terms allows us to reproducibly identify the endpoints relevant to the KCs and to reproducibly across different agencies, industry groups, nonprofits, to come up with the same reproducible collection of studies to be used to evaluate the KCs. And that makes all of our efforts kind of more trustworthy, right? Another thing that we can do beyond the actual search is that this helps us organize the gathered evidence and integrate it. So for example, when I worked with the IARC monographs to identify carcinogens, I was assigned reviewing all of the mechanistic data for KC number 4. And in this instance, it gives the person or the organization that is delegating the work of evaluating the KCs, a way to delegate the work in a manageable way for the team that's working on it. And so you can then bin all of this information you've systematically and transparently gathered into concepts that relate to these 10 KCs, and then bring everyone together to integrate this gathered evidence on endocrine disruption. And you can do that across the data streams. So not just across the KCs themselves, but you might also have integration with the epidemiology studies and the whole animal studies. So one of the things that's great about the KCs is it will help you identify molecular epidemiology studies, which can, for example, help us see whether or not a mechanism appears to be operating in human tissues or cells or bodies. We can also integrate across in silico models or high throughput tests that are becoming more common in some of the regulators' toolkits. So one of the tricky things, or as I call it here, kind of a hazard of the relevant literature, is that depending on what a given group considers relevant literature, you might have a lot of missing data when you go about looking for evidence to support identifying an EDC. And so to illustrate that point, here's a table from the article where we're looking at the guideline standardized test by the US EPA as well as the Organization for Economic Cooperation and Development, the OECD. They have a number of kind of official standardized tests that they like to look at to evaluate endocrine disruption. And what you can see on the right of this table is that they have tests in position to address KCs 1, 2, and 6, but the other KCs do not yet have standardized tests. So if these organizations were working with this framework, they could still work with the framework and benefit from its ability to organize in a transparent way, but they would probably be missing some of the aspects of the characters of these EDCs. And so probably some chemicals that are truly EDCs are not being called EDCs by the OECD and the EPA because of these missing KCs and their standardized test portfolio. But one thing that's really kind of exciting in terms of being an academic researcher or someone who's interested in developing assays is this is really, I think, a call to action of let's develop better assays that are standardized to populate these kind of missing KCs here. So another thing that we can do is something I kind of touched on before, and because we don't have to do a system of drawing arrows between one piece of evidence to the next to say how, for example, receptor binding explicitly relates to a key signaling event or a hormone synthesis or clearance, that because you can look at these as the characteristics, you're not forced to describe an entire specific molecular pathway. And as I mentioned before, that's a very hard bar to fill, even with the well-known hazardous chemicals. We struggle as a scientific community to do that. And because we can combine these key characteristics with phenotypic data, both from our human studies and our animal studies, this strategy really represents an important and very practical addition to making our way through hazard identification in the world community. So I'd like to give you a quick case study to illustrate some of these points, and that's with bisphenol A. So probably many of you have heard of this. This is a short-lived but ubiquitous exposure. It's commonly found in plastics and also thermal paper receipts, among other consumer products. So we did a kind of thought exercise because a number of the members of the consensus team are internationally renowned experts on BPA without having to come up with all the search terms and formal transparent search, if you will. We searched our brains, okay? And what we concluded about the humans is there are now more than 100 epidemiology studies that are showing positive associations between bisphenol A and adverse endocrine-related outcomes such as obesity, diabetes, and particularly type 2 diabetes, female infertility, male sexual dysfunction, lower birth weight, and even atypical neural behaviors. Okay, so many of the studies that have been conducted in humans are cross-sectional, meaning the same time that the BPA was assessed, the adverse outcome was assessed. But other of these studies are longitudinal, meaning we looked at the exposure before we looked at the bad outcome, and that helps us have a stronger confidence in that data. So people have, I'm going to focus now on some relationships between bisphenol A and type 2 diabetes. I've used this as an example in part because this is an area of research generally that I conduct work in. And what I'm illustrating to you here is an example of a rigorous study that was done where you use analytical method called meta-analysis to look formally at whether or not across all the available human studies, whether or not there's a relationship between bisphenol A and type 2 diabetes. And in that study, they did find that the majority of studies found that there was a relationship between bisphenol A and an increased risk of type 2 diabetes. So that's the part of the evaluation that represents looking at the human data. Now let's shift gears to say, what do we know about animals? And so you can see in the diagram above, I've got that bolded. So in rodents, there's also been hundreds of studies, and they're demonstrating that low doses of BPA can disrupt the development of a lot of those tissues that we were just talking about in terms of those diseases, specifically the brain, reproductive systems, including the mammary gland, and various what we'd call metabolic tissues under endocrine control, such as the pancreas, which produces insulin. We also have seen evidence that BPA can also include precancerous and cancerous lesions, so tumors of the mammary gland and the prostate. So by doing a full data analysis of what we know, we can say these things about the animal phenotype. So now let's shift gears and look at the mechanisms. Okay, so we're doing this thought exercise about how do we look agnostically at the literature of all the mechanistic studies for BPA and put it into the KCs in order to get a better picture of how that evidence may support some of the phenotypes we just talked about in the humans and animals. And so what I've done here is put all that data into a table that's divided into 2 slides for each characteristic. And because we have not a lot of time today, I'm highlighting the specific parts that relates to that diabetes example. Okay, so I will touch in the overall discussion a little bit of some of these other features, and I hope that while I'm speaking, your eyes are ability to go ahead and read over this. I think one thing that's very clear is that there's a lot of evidence for BPA in each of the key characteristics 1 through 5. And in particular related in an obvious manner to diabetes is that BPA can repress pancreatic beta cell calcium signaling. So I know that's a mouthful, but that's really under the signal transduction KC4, calcium signaling is what happens in our pancreas right before insulin is secreted. And so this is a really easy one to understand why that would be related to the diabetes association we discussed. You'll note on 1 that there's also BPA activates nuclear ERs, those are estrogen receptors, and membrane ERs. And in number 3, also the expression of the estrogen receptor. And in number 4, also the signaling of the estrogen receptor. And I'm bringing that up because it turns out that the estrogen receptor actually talks to the insulin system. And we'll get into that in a little bit. So let's look at the second half of that table. So similarly, you can see that BPA has mechanistic evidence for most of these EDC characteristics. You'll see here at number 9, we've named no evidence. It doesn't mean that there is never going to be evidence or that it doesn't yet exist today, but among our expert team, we were not aware of it at the time of this publication. And I think it's really important to understand that even though there are 9 KCs of EDCs that there is evidence for BPA, the KCs of EDCs are not simply something to just be counted up. And so it doesn't matter if there's 9 or 10 or even just 1. Here you can see in bold number 7, I mentioned the insulin secretion. We can see how you could potentially connect the dots between these KCs, because after we have that calcium signaling that I highlighted, as I mentioned, you have insulin secretion, and lo and behold, when you look at insulin secretion, you do see that low BPA doses increase that secretion. And so that's changing the hormone transport across cell membranes. It's getting it out of the pancreas and in the bloodstream where it can go do what it wants with its target tissues. Now, that means that even if you really want to draw arrows, you could still use this framework to help you organize the data in a transparent way. And then if you want to put the extra effort into taking a table like this and making a picture with arrows, you could. But you can see here that you're already getting a lot of information just by tabulating. Now, number 10, I mentioned, is important for differentiation. What we're also seeing that I'd like to highlight is that BPA increases the proliferation index in the pancreas, suggesting that the pancreas is reproducing its cells at a higher rate. So what happens in diabetes is insulin action starts not working as well, and the pancreas starts making more insulin and producing it. So you have that increase in insulin secretion. Because of this, it kind of wears out the cells faster, and you actually have the need to produce more cells to try and compensate. And so you're seeing here evidence that's consistent with a kind of pre-type 2 diabetes state. So I'd like to just show you that data very briefly. And what you can see here is that BPA increases insulin secretion in mice. This is a dose response of BPA on the x-axis, and you can see the insulin coming out of the pancreatic islet when it's stimulated by glucose, which is how you get insulin to come out of the pancreas, because we secrete it after meals. Now, the authors went one step further and actually showed that this relationship seemed to be related to estrogen receptor beta signaling, because of when they made it so that the the pancreas cells didn't have the insulin receptor, the estrogen receptor, that we no longer had the increase in insulin secretion. So when you get rid of a receptor and the action doesn't happen, that means that the receptor is important for the action. And so how on earth is estrogen related to insulin? I'll just leave you with this idea that the authors came up with. So you can see that sometimes, yes, we're going to draw arrows, and they are helpful in us understanding. You can see at the top left, bisphenol A is activating estrogen receptor, and this is changing some signaling, including calcium release, which I told you when we get more calcium, we secrete that insulin, okay? So upstream of insulin secretion, we have estrogen receptor signaling and activation. And so what you can see is, if we integrate now in an overall evaluation some of the items that I've just touched on, we've really got thousands of studies that are supporting 9 of the 10 KCs evidence for the BPA. And just to summarize what I think is key for the type 2 diabetes part of the story is that BPA both binds and activates the estrogen receptor, so that's KC1 and 2. And then through KC5, we have seen separate studies looking at epigenetic modifications that help the estrogen receptor be activated. We also see the signal transduction, so say KC4, consistent with the above events that I just mentioned. And through that signal transduction, there's a couple different arms that are occurring. One, you can see in the other part of the diagram that KC6 is mediating insulin transcription, so meaning it's increasing the amount of insulin, and also it's activating KC7 or embodying that, where you're getting that insulin secretion or hormone secretion. So these mechanisms are consistent with the diverse adverse effects we're seeing of BPA in humans and in other animals. And in this example, that relates to type 2 diabetes, but I'm sure you all are well familiar with ER's important role in the reproductive system, including the mammary glands. So with that, I would really like to thank all of the EDC group participants. This wouldn't be an international consensus statement without the international team that we worked with diligently for over a year to refine these concepts. So I'd like to personally thank Dr. Brown at the OECD, Dr. Coliano, who is at the US EPA, Dr. Goodson in San Francisco at the hospital, Dr. Goiten at the IARC, Dr. Korach at the NIEHS, Dr. Kortenkamp at Brunel in the United Kingdom, Dr. Rizwick at University of California at Berkeley, as is Dr. Smith, Dr. Sone, who's at the National Institute of Environmental Science, a kind of NIEHS of Japan, Dr. Vandenberg at University of Massachusetts, Dr. Woodruff at UC San Francisco, Dr. Zeiss at the CalEPA, who also asked for this work to be conducted, and finally my key partner, Dr. Zoeller, also at UMass. So thank you for your attention.
Great, thank you so much, Dr. Merrill. Now it's time for our Q&A session. You may type in your question through the Q&A feature available on the menu bar at the top of your window. We will get to as many comments and questions as we can. Jerry, do you want to take the first question for us?
Okay, thank you. So the first question is, have you or your co-authors talked to the European Union about using the KCs in their EDC criteria, which requires showing a mechanism is linked to the adverse EDC effect?
So thank you for the question. I personally have not talked to the European Union. I have given a presentation to the OECD about this consensus framework, and so they're well aware of it. And members of the European Union, of course, were co-authors on this, and other people who are working on endocrine disruptors in the European Union are aware of this publication. So I'm under the impression that the EU has also been made aware of it in terms of their endocrine disrupting programs, but I don't have any direct information I can share about the nature of that.
Okay. So one thing that kind of comes to mind is if there's 10 KCs, if a chemical affects more KCs, does that mean it is a stronger endocrine disruptor or something that's more important than one that affects just one or two?
Right. So one of the things that IARC has found, having implemented the KCs of carcinogens already, is that known carcinogens often have several of the characteristics embodied. However, you need not have a majority of key characteristics or even half of them. So some chemicals may only have evidence for one or two. So I don't think it's appropriate to really just count the number of KC hits, if you speak, in order to decide about the strength of evidence. There are a number of organizations that have put a lot of academic thought into how do you decide about strength of evidence. And often it comes back to how the study qualities are evaluated. For example, did they blind the selection of which groups of cells or animals went into treatment groups. Did they have a sufficient N? Did they do appropriate statistical analysis and so forth? And so one of the uses of these KCs is then to be kind of have the data fed into, if you will, these frameworks that look at the strength of evidence. IARC themselves also looks at strength of evidence when they gather the KCs of carcinogens, and I think that that would be an appropriate way to rigorously evaluate whether or not you've found evidence for one KC or all 10.
Okay, kind of the last question. So there are 10 KCs, but are there examples for all 10? It seems like there's, you know, some of the bottom ones, there may not be any actual examples right now. Is that true?
Do you mean for the standardized tests?
Just examples of EDCs that affect that key characteristic.
Well, BPA is hitting 9 out of 10 of them. In our paper, we also give example for DES, the pharmaceutical that's a estrogen mimic, and that one I believe we have all 10 mapped to. I also conduct a lot of work myself on DDT and DDE, and I have mapped DDT pesticide and its metabolite DDE to all 10 key characteristics. So I think as this gets implemented, we'll see a lot more examples of all the different contexts in which you can have 1 or 2 or 9 or 10.
Okay, great. Thank you very much. So let me turn it now back to Hannah.
Great, thank you so much, Dr. Merrill and Jerry. We're approaching the end of our webinar today. A video recording of this webinar will be available on the CHE website soon. Tomorrow you'll receive an email containing a link to the recording. If you are new to CHE and would like to stay updated about upcoming events and more, please sign up to receive our newsletter by selecting the Join Us tab at the top of any page on our website. Additionally, if you appreciate the CHE Partnership webinars bringing you the latest environmental health research for free, we encourage you to support CHE's ongoing work by making a tax-deductible donation via our secure website. Again, our website is healthandenvironment.org. With that, I would like to thank you, Dr. Merrill, again for taking the time to talk to us today, and to Jerry for your excellent moderation. Thank you for joining us, and have a good day.